right? You do not need to explain, okay? But if the question asks to elaborate, right? Elaborate means that you need to explain. Okay? Understand? Understand? Look carefully at the question. Alright? Another word that you need to explain is justify. Okay? So let's say you are given Gibbs energy delta H minus T delta S. Okay, we know this is Gibbs free energy, right? I will not give this in the examination. You need to memorize the equation. Okay, so let's say you calculated the value, and the value, right, is negative or positive. Right? The question will ask, justify your answer. So from this answer, you need to explain. This negative, what does it mean? Does it mean exotonic? Then you need to write. If it's negative, then you write exothermic. Right. So if exothermic means heat is released from the system, right? Heat is being released from the system. So what happened? So system is hot, or you can say heat is released from the system. So this process, exothermic, is it spontaneous or not spontaneous? Spontaneous, guys, or non-spontaneous? Spontaneous, right? So exothermic, system is hot, heat is released. This process is spontaneous. That's simple. So you need to justify based on your answer given. Okay? So a few words that you need to take note of. Alright? So if you have words such as provide, alright? Provide. What else? Provide. Provide is also similar with state, meaning that you only give examples, you do not need to explain. Okay? If the question asks draw, okay? Draw, it means that you need to draw the mechanism. Do you still remember the quiz we have? The quiz, there is one question, you need to draw the amino acid. You still remember? You need to draw the errors. Okay? What happens at the error? Okay? How many electrons is being donated? Okay? Remember? Alright. So these are the few keywords that you need to take note of. Alright? These two, right? You need to explain. State, provide, this means you do not need to explain but provide the examples. If the question asks draw mechanism, you need to draw the mechanism. Okay? Okay. So chapter 1, coming back here, chapter 1, what do you need to focus? Because chapter 1 we talk about prokaryotics, eukaryotics. A lot we mentioned about the organelles, which is this slide. Okay. So in the organelles we have cell wall, right? We have cell membrane. We have also different kinds of organelles such as endoplasmic reticulum, we have rough endoplasmic, smooth endoplasmic, right? We have ribosomes, Golgi apparatus or Golgi bodies, and these are microbodies, okay? Mitochondria, chloroplasts, okay? So, in this, all of the eukaryotics function, okay? Please, I will pick a few. I will pick a few of the organelles and I will ask you the function, okay? I will ask you the function. I will not let you know, oh, I choose chloroplast, then tell me the function, okay? I will choose from here, then you all need to know what is the function, okay? Clear? And then specifically on Golgi bodies, right? What is actually Golgi bodies? When you only state the function, right? State the function, right? What is the function of Golgi bodies? How do you need to explain, right? So how do we explain that thing? So Golgi bodies or Golgi apparatus function to packaging, right? And also transport of all these proteins and lipids into microbodies. Okay? So you need to explain this, alright? Okay. So another one. How many types of microbodies there is? How many types of microbodies? There are 3 types, okay? Lysosome, perioxomes, and glyoxosomes. Okay? So please take note on these 3 types of microbodies. Okay? You need to elaborate what are these microbodies. Okay? For example, if question asks, justify, right? Justify these 3 types of microbodies. So what does the first microbodies do? Hydrolyze the protein into hydrolysate, lysosome, lysosome. So the first one, lysosome. And this function as hydrolysis. Okay. So again, you need to write, instead of writing lysosome, alright, I give an example. You need to write carry out hydrolysis process to carry out hydrolysis process to obtain hydrolysis. Okay? Instead of writing this only, right, you need to write this as well as the explanation. That is what I meant. Okay? If the question asks to justify. Alright? Okay. So if the question asks about what is responsible for photosynthesis process, okay, state or elaborate, the word again, elaborate. Elaborate, what does it mean? It means that you need to further explain, okay. So if the question asks to elaborate 2 stages of photosynthesis, so you need to explain what are the 2 stages of photosynthesis. The first stage What is first stage? Light dependent. What does it mean by light dependent? Do not just write light dependent. You need to explain. Light dependent is actually requirements of light in this stage. Okay, meaning that it requires UV with the carbon dioxide in order to form what we call sunlight is captured through chlorophyll and carotenoids convert into ATP, right? Process known as carbon and convert carbon dioxide known as carbonification. Okay, and this is formed into forms of ATP plus and ADPH. This is in the forms of energy, the first stage, right? Right? So the second stage, then you need to explain light independent, right? It means no sunlight, you do not need sunlight. So what happened then? Because of the energy received from the system already, okay? So what happened? The light independent, also known as Calvin cycle, electrons carried by NADPH converts carbon dioxide to carbohydrate. It means that perhaps the CO2 get one of the phosphate group energy, right? Therefore convert into carbohydrate. Okay, and this carbohydrate is actually a form of energy. Okay, so this is the easy one that I'm giving you, right? But you need to explain, okay, clearly on this, right? Okay, chapter 2, part A, okay? Understand? Understand? At the back? Everyone okay? Chapter 2, part A. Clear? Everything about the cells, organelles, okay? And a little bit about what is the reaction, okay? Chapter 2, part B. So chapter 2, part B is about the chemistry of water, okay? So please know how to differentiate between covalent and also non-covalent, okay? So once you know how to differentiate between the covalent and non-covalent, the next one you need to take note is on this part, solvent properties. Alright? So if question asks, again, the word elaborate. Okay? Elaborate what happened when water dissolve a solute. Okay? A water dissolved solute. Okay? This part. What happened when the water, which is solvent, dissolve the solute? Okay? So please take note on the keywords. Once they interacted, what happened is that they create dipolar interaction. Okay? So what happens to the dipolar interaction? Meaning that they have Na plus Cl minus interacting with H plus and OH minus of the water. Okay? So what it creates? Covalent, right? Covalent, which is a hydrogen bonding. So the hydrogen bonding from the water will interact with the sodium and also the chloride. then it forms strong electrostatic interaction and then creates hydration shell. Clear? Okay? So this is what you need to take note of, the keywords, alright? The keywords that are highlighted in green. Okay? Alright, the keywords that you need to take note of. Okay? So, if that question asks, what is actually the differences, right? What is actually the differences between hydrophilic, hydrophobic? What is the differences hydrophilic, hydrophobic? Then you need to explain what is hydrophilic, hydrophobic. Okay? Or if the question asks, what happens when water interacts with a non-polar solute? What is a non-polar solute? Non-polar means no charge. Okay? No charge. So what happens? There's no interaction, meaning that it creates hydrophobic interaction, because non-polar is a non-charged solute, water is a charged solute. So there's no interaction that will occur during the reaction. Okay, so create hydrophobic interaction. Right? Okay. So, from the chemistry of interaction, we know there are two types, covalent, non-covalent, right? So please again, take note on this. Covalent means sharing a pair of electrons to form a strong bond between atoms, right? Right? A, B, right? Shares one electron, two electron, they share, forms covalent, right? And this bond is very strong compared to non-covalent. Okay? So non-covalent, what does it based on? Electrostatic, right? Positive, negative charges, right? And this is what we call ionic interaction. Or it can base on partially positive or partially negative charge, right? What else? Hydrogen bond, hydrogen H plus, hydrogen bonding, okay? And then we have hydrophobic that I've explained. That is no charge, right? Hydrophobic, right? There's no charge between solvent. And if we talk about hydrophilic, we know that hydrophilic means water-like, so it's usually OH minus H plus, this kind of charges, right? Okay. The last one is van der Waals. Right, van der Waals. So what is van der Waals? Right? Van der Waals has these three charges: dipole, 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 induced dipole, induced dipole, induced dipole. So what does it mean? Dipole means charge, right? Another one, induced dipole. Induced dipole means no charge, right? So there is this kind of interaction happening. Okay? Okay? 可以吗 Clear? 后面的 OK? Clear? Suddenly the class full attendance today. Even I come for 1 hour, the class full attendance because the tips. Okay, anyway, Okay, because we talk about hydrophilic, hydrophobic, there's another one, amphipathic. What is amphipathic? Amphipathic means combination of hydrophilic and also hydrophobic. For example, right? Right? This is polar. This is non-polar. So this carboxylic acid is one of the molecule, right, that is amphipathic. Consists of polar, non-polar. Okay? Alright, this is what we meant. Hydrophilic, hydrophobic characteristics. Okay. We have some time now. Please take note on this. Osmotic pressure, if the question asks to elaborate osmotic pressure, three types: isotonic, hypotonic, hypertonic. Okay? So what is the flow of isotonic is equal. Okay? What is the flow of hyper? From out to in or in to out. Alright? Then you need to explain what happened to the cell. Okay? This is what we meant. Hypotonic, if the cells, water outside going into the cell. So what happened to the cell? The cell will explode, right? So the process of the cell expand and will create lyse. Please write the keyword, expand and lyse. Hypertonic meaning that water inside the cell goes out, right? Hyper, the cell inside goes out. So what happens is that water particle will move out the cell, causing crenation. So what is crenation? Your cell will shrink, right? Right? Because water comes out from the cell. Okay? So please explain all this if the question asks to justify. Okay? If question like this asks for buffer system, then you need to explain what is actually a buffer. Okay, buffer helps to regulate or maintain the hydroxide or the hydrogen ions in a solution so that the pH will maintain. All right. Okay, so this is the last. I think this is the last one. Oh no, not yet. Okay. Okay, gives free energy. This one. Alright. Please take note. I think I share with you guys this one. So again, gives free energy. The equation will not be given to you. You need to memorize the equations. Okay, I think it's very simple. So question will give something like this, I think, right? Then the enthalpy value delta H will be given, right? The temperature will also be given. Check nicely the question, okay? Whether is it 30 degrees or perhaps in the room temperature. If it's in room temperature, what is the value? Room temperature. Guys, what is the value for room temperature? Guys, room temperature 23, 24, 25, 25, okay? Room temperature is 25, universal room temperature, okay? So, check carefully the question and then put in the value, alright? If the question asks for room temperature is 25, okay? Don't look for another temperature, 30 or other temperature than that, okay? 25 is room temperature, alright? So substitute substitute all the values that are given in the question asked into Gibbs free, and then explain. Okay, explain. If it's negative, this is how you need to explain. If it's positive, this is how you need to explain. Okay, clear, easy. Explain, explain. All right. Okay, because we have understand what is endergonic and what is exergonic. Okay, from this slide, right? Exergonic, endergonic, right? Heat is released, heat is absorbed. Okay, so if the question asks, provide example of exergonic, provide example for endergonic. So what is the example? Okay, for example, when we breathe in, we exhale of carbon dioxide and water vapor. Okay, what is the process? Exergonic. Okay, so write down the equation accordingly. The second one, ATP to forms ADP. This is endergonic reaction. Okay, so give examples what is exergonic and endergonic. Okay, the one that we have discussed before, photosynthesis process like this just now, carbon dioxide, UV to get oxygen and water. This is an endogonic process. ATP synthesized, okay, the reverse process to get back ATP. Energy is being absorbed. This is also endergonic process. Okay. Okay. So I think these are all the things that you need to take note of that will be covered for this. Okay. So that is for question 2, question 3. Okay. So chapter 2 part B, you have 2 questions. Chapter question 2 and also question 3. Right? Clear? So 1, 2, 3 done. Okay, settled already. Right? Right now, question number 4, which is on amino acid. Okay. So coming back to amino acid. So coming back to amino acid. You need to understand the structure of amino acid. Okay? Very simple. This is amino group. Okay? This is carboxyl group. Okay? You have the H group, right? H. But basically this will not change, okay, the hydrogen. Most important is the R group. How many R group do we have? 5 R group. Polar, non-polar, aromatic, positively charged, negatively charged, right? 5 groups, okay? So if question asks, right, how to actually identify the classes of amino acid. So that really depends on the R group. Okay? Depends on the R group. Alright? Let me go straight to the point. I think we have discussed all of these, the different types of categories of amino acid. Right? non-polar, polar, aromatic, positively charged, negatively charged. We have looked into each of the amino acid groups. Okay. Let me go straight to the point. Okay. So if question asks to draw the mechanism, okay, please take note that the arrows is very important. You do not need to memorize the structural, okay? I will not ask you to memorize all the 28 types of amino acid. You do not need to memorize that, okay? I will give you the structural of the amino acid. What you need to do is, you need to draw dipeptide formation, okay? So how to draw the mechanism? Okay, this is how we draw it. So make sure a few things. The electrons, the arrows, okay? The electron as in +1 or -1, please write clearly. This one, please write clearly all the arrows, okay? The +1, -1, right? Okay, which is a good living group? Good living group, H2O, leaves the water molecule, the one that I've highlighted. Please take note on this, okay? All the arrows. This I think around 5 or 6 marks, 5 marks or 6 marks, okay? So which attack which, and how does the peptide bond is being formed, okay? At this part, okay? So please take note on this, okay? All right, clear? Okay. Please make sure all the arrows must be drawn. Okay. Right. So I think we are done for this mechanism. So you need to know how to draw the mechanism. Okay. So after drawing the mechanism, not just drawing the mechanism, please remember to give name of the dipeptide. If the name of the dipeptide is lysine, then just put accordingly LYS, and then with serine, okay, SER, okay. This you can get the mark as well to give a simple name, right? What else? Another part which is to calculate the pH value, okay? To calculate the isoelectric point, right? How to calculate isoelectric point, right? I think we've done this, right? In the class we have done this. Check the isoelectric point from the R group, which is the highest or which both value is the lowest, and then that is the pH. And then from the answer that you get, right, you can justify your amino acid, whether is it acidic or whether is it basic. Okay, so please take note on that kind of question. And the last one is, what are the two types? I think I mentioned about this. What are the two types of amino acid that contains sulfur group? What are the two types? Do you still remember? Okay, go figure it out. Okay, what are the two types of contains sulfur group? Okay, there's two types of amino acid that contains the disulfide sulfide groups. Okay. Okay, so I think that will be all for amino acid. Okay, just take note. Maybe the small question will be like, what is zwitter ion? What does it mean by zwitter ions? Okay, Okay, so I think that will be all. Still have 5 minutes. Okay, proteins. Right? So please take note on this part, architects of proteins. So how many levels we have of architects of proteins? 4 levels, right? Primary, secondary, tertiary, quaternary, right? We have go through each of the levels of proteins. Okay? So for primary, what happens in linear sequence? It means that they are bound by covalent bond from amide or peptide bond. Okay? The second one, right? What happened? Right? The third one, right? The covalent and non-covalent interaction happening. And what is the fourth? Quaternary. Okay? And then in chapter number 4, I think we have discussed the different types of proteins. There are many different types of protein. Structural proteins, right? Regulatory proteins, exotic proteins. You need to know how to differentiate this kind of protein, which is fibrous, which is globular. Okay, which is fibrous, which is globular. Right? I think we have looked into this. Fibrous, globular. So fibrous, it means that it has a lot of roots, right? So this is hydrophobic. Globular, it means it's round, therefore is hydrophilic, right? Okay, so we have looked into all these type of different proteins last Thursday. Okay, so from these different kinds of proteins, I will give maybe 2 or 3 types. Okay, let's say, let me give you an example. Let's say if I give you structural proteins. Okay, first look at the keyword. If you need structural proteins, fibrous or globular. Structural proteins is fibrous or globular. 还记得吗? Which one? A or B? Structural proteins, A or B? A. Guys, if A, please raise your hand. Fibrous. 后面的拿住手机的 for what? A fibrous, okay. B, okay. 没有举手呢? Okay. Please take note on this, okay? Giving, when you provide already, you state, oh, structural is fibrous, okay? Then what is the functional of structural proteins? What is the function of structural proteins? Okay, you state it provides structural strength to the cells, and then you need to give example. What are the examples of structural proteins? You can see here collagen. Then you can put collagen, keratins. Okay, clear? All right. Okay. The last question. Okay. The last question is the separation process. Okay. I will give you the graph. Alright. And then you need to justify which graph is corresponding to the types of protein characteristics. Okay? So which one you need to check? the stationary phase, right? The stationary phase, whether is it polar or non-polar. So check back the question. This question will state very clearly. Stationary phase is hydrophilic or stationary phase is positively charged. Check the question, okay? And then you can identify, right? Same charge, repel, right? Different charge, attract. Hydrophobic and hydrophobic will attract. Then it will come out which is hydrophilic, okay? So check the stationary phase, okay? The stationary phase is very critical at this part, okay? This one. Alright. So after you already mentioned, oh, this is non-polar, or this is polar. The second one again, why? Justify your answer. Why? Okay? Justify why polar, why non-polar. That's all. Okay? Alright, so I think that is all for your midterm examination tips. Okay? So I think I've covered 3 chapters. Chapter 2, part A, part B. Question 1, 2, 3. Chapter 4, right, is for Chapter 3 is for question number 4, amino acid, on the mechanisms, on the pH isoelectric, alright, on the amphipathic, or what are the types of R groups. The last one is this one, proteins, okay? Architectures of proteins, what else? Fibrous globular proteins. The last one is on this one, separation of proteins. Okay? Alright? Okay, so that is all for the midterm.